Hi and welcome to Learn Biology with Becky. This is another walkthrough of a multiple choice paper. This was from May-June 2019, so the most recent paper sat in the UK area. As usual, we're going to go through each question and give a full explanation of the right answer. If you have any questions, please get in touch. Please subscribe to the channel so you can access more of our walkthrough videos. Thank you and enjoy. Okay, so question 1, we have a diagram. I'm just going to zoom out slightly so that you can see the whole question, which would be helpful. All right, so question 1. This diagram shows a leaf on a plant. We have the sun shining on the leaf, and then it's showing that water is coming into the leaf from the soil. Carbon dioxide comes into the leaf from the air, and simple sugars are made in the leaf. The question is, which characteristic of life is represented by this diagram? Now, as I said in the previous walkthrough, often they have a Mrs. Gren question as question 1. So they're looking for a characteristic of life. There's movement, respiration, sensitivity, growth, reproduction, excretion, nutrition. So of the answers, they are all a Mrs. Gren characteristic. But what is this actually showing? Well, it's actually showing photosynthesis being carried out, which is how the plant gets its nutrition. So the answer is B for question 1. Next question also has a diagram, a flow chart. It says the diagram shows how Homo sapiens, also known as modern people, could have evolved from earlier ancestors. And the flow chart showing evolution from Homo habilis and through to Homo sapiens, the modern people, which we belong to. So which statement about modern people and their ancestors is correct? A, are they in the same species and the same genus? Well, if you look at the name, it's what we call a binomial name. So there's two parts to it. This first part, which has a capital letter, is the genus name. So that's Homo. So they're actually all sharing the same genus. However, the second part of the name is the species name or specific name. And that's actually starts with a lowercase letter, as you can see. And that's different between the 4 individual species that you can see from the bottom through to the Homo sapiens. So we're looking really for same genus but different species. So if we go through the answers, A, they are in the same species and the same genus. Well, that can't be the case because the second word, the species name, is different as we go along the flow chart. They are in the same species but not the same genus. Well, no, the species is different. It's the genus that's the same. So just watch wording because these are very wordy questions that are very similar. They are in the same genus but not the same species. Now that one sounds good to me, but let's just check D. They are neither the same species nor the same genus. Well, they've got the same genus name, so D is not right. We're going to go for C. Question 3, we have a photomicrograph. So that's just a photograph taken via a microscope, and it shows part of an animal's cell. So this is quite a magnified image here. It says, what is the structure labelled X? And the options are A, chloroplast, B, mitochondrion, C, ribosome, D, vacuole. Now, ribosomes are very, very small structures. They almost look like little dots really in a cell where proteins are made. So that doesn't sound right. The vacuole is the sort of central part of a plant cell, and it doesn't tend to have any inner membrane like this structure has. So we will cross out vacuole. And then that leaves us between a chloroplast and a mitochondria. Now, chloroplasts, which are found in plant cells, big clue, chloroplast plant cells, just watch any information given. They've told us it's an animal cell. So straight away, we can say it's not a chloroplast. So that leaves us with B, the power of elimination. It's the mitochondria. And mitochondria do indeed have these membranes, these inner membranes that sort of project inwards into the cell structure. Question 4, which features are possessed by all, if it's in bold, take note of it, plant cells? So we're looking at just whether it's got a cell wall or chloroplasts. Now, all plant cells have a cell wall, so we can eliminate C and D, get them off the table. So we're just looking at A and B. That leaves us just to look at chloroplasts. Are chloroplasts present in all plant cells, or are they not present in all plant cells? There are quite a lot of plant cells that have absolutely nothing to do with photosynthesis. Imagine all of those cells underneath the soil, the root hair cells and so on. They're never going to get any light, so they do not have chloroplasts. So B is the correct response. Number 5, the diagram shows part of the skin. So you should have learned about the structure of the skin as you were covering probably the nervous system is where it would come up, or maybe homeostasis, depending on how your teacher taught it. It says, what is the correct level of organisation for the skin? So is it a cell, organ system, or tissue? Well, straight away, there's absolutely no way it's a cell. Okay, the skin is a very complex, it's actually an organ, it's very complex. You've got hair, you've got oil glands, you've got blood vessels going in, here's a capillary, you've got sweat glands, you've got nervous tissue. So actually, and also, sorry, down here, you've got the fat tissue as well for insulation. So actually, it's made up of several types of tissue, so we call it an organ. And it's not an organ system because that would be several organs working together. So actually the skin can belong to the nervous system for responding to changes in environment, like change in temperature, or it could be part of homeostasis because it regulates our body temperature and, you know, carries out things like sweating and so on. Number 6. The diagram shows a test tube containing clear jelly. A drop of blue ink is injected into the middle of the jelly. And there we have a picture just to help us visualize that. It says the blue colour of the ink spreads throughout the jelly. By what process does the blue ink spread through the jelly? Now, the ink, the coloured pigment, it's going to move from where it's more concentrated to where it's less concentrated. And that process is known as diffusion. Don't be tricked into putting osmosis as your answer. Osmosis is about water moving, whereas we're actually talking about the pigment, which isn't necessarily water. It might be dissolved in water, but it's actually the blue pigment, the chemical, that's moving or diffusing through the jelly. Right, number 7. Okay, we have a diagram, a setup of apparatus. So just take your time to work through what's going on. Pure water in the beaker. And then you've got a glass tube, and in the glass, at the end of the glass tube, is sort of bulb-like terminal to it. We've got a sugar solution with blue ink, and there's a partially permeable membrane here at the base of the glass tube. It says, some hours later, the water in the beaker had turned blue. So this water here had turned blue, and the liquid in the glass tube had moved upwards. So it's actually higher up in the glass tube. Which process caused these changes? Now, first of all, it's a bit repetitive from question 6, really. The reason why the water has turned blue is just as you answered in the question before, it's down to diffusion. So that leaves us with C or D. So remember, elimination is your key kind of skill to get through these papers and help you focus on the correct answer. Let's get rid of 50% of the answers, leaving us with C and D. Now we're looking at what's happened to move the kind of level, if you like, upwards, is that water has moved in from the pure water to the sugar solution. So they're trying to confuse you because there's two different things going on here. So the second part of the table is that water's moved in and it's gone from where it's at a higher water potential, where there's no solute, there's no sugar, to where there's a lower water potential due to the sugar being present in the water. And the movement of water molecules from high to low water potential, of course, is osmosis. So in your table, we need to get the right combination. We already decided it was diffusion, but for liquid in the glass tube moving upwards, it's osmosis. There's no way active transport could take place. This is not living material. It's just probably made up of a bit of visquing tubing and some glass. Active transport would require some way of releasing energy, and it would only happen, obviously, in living cells through respiration. Okay, number 8. 4 solutions were tested to see whether they contained protein, starch, or glucose. So straight away you should be thinking, this is my food tests or biochemical tests as they're correctly known. It says the colours of the solutions after the tests are recorded in the table. Which solution contained protein and glucose but not starch? So first of all, to get anywhere with this question, you need to know which test is testing for what. So first of all, the Biuret test is the one we use to test for protein. Glucose is the Benedict's test, and starch is the iodine test. Now we can go at this different ways, but it might be easiest just to look for the one that's showing no starch. So the iodine test will only go blue-black if there's starch present. Now we're saying there's no starch, so any of the rows with blue-black underneath the iodine test, they need to go. We need to eliminate them, and that leaves us with A and C. Now it says the solution contained protein. The Biuret test, if there's protein present, will go purple. whereas A is showing it stayed blue, which means that no protein is present. So C is our answer. We haven't even had to look at the Benedict's test. We haven't even had to look at all the bits of information. We've managed to eliminate all the other three and work out that C is the correct answer. But just to be sure, if we look at the test for Benedict's, that wouldn't have helped us anyway because it was the same answer. It was orange for both A and C. So it was only the Biuret test that would have allowed us to eliminate A and to find that C was the correct answer. Okay, number 9. Small molecules are used as the basic units in the synthesis. That's just a posh word for making large food molecules. Which statement is correct? So amino acids are basic units of carbohydrates. No, they are the basic unit of a protein. That's going. Fatty acids are basic units of glycogen. There's quite a few words that begin with G that are very easy to confuse in biology. Glycogen, glucose, there's hormones like glucagon. So it is, and glycerol, which we'll come to in a minute. Fatty acids are used to basically make lipids, okay? Now, glycogen is actually a carbohydrate. It's how we store our glucose in our body. So fatty acids don't have anything to do with carbohydrates, so B is incorrect. Glycerol is a basic unit of oils. Yes, oils are a type of lipid. They're a lipid that's liquid at room temperature. And glycerol and fatty acids together would make a lipid. So C is correct. D, simple sugar is a basic unit of protein. Absolutely not. Simple sugars are a carbohydrate. So C is our correct answer. Question 10. The diagrams show a protease enzyme catalyzing the breaking of part of a protein molecule into smaller pieces. So effectively, this is what happens during digestion. Which diagram has 3 correct labels? So it might be a bit overwhelming because there's lots of similarities going on here, but it's a process of just elimination with multiple choice. Don't forget that. Let's go through. With A, we've got the product. Okay, the product, that doesn't seem quite right. It's just kind of in the active site, so it's not been released yet. The product is what's released from the active site, which is kind of here, these 2 smaller parts. Enzyme, yeah, it looks like an enzyme to me. It's got a little cleft, what's known as the active site. And substrate looks right to me. It's this bit I'm not so sure about, so we'll move on. We can always come back and, you know, like I say, if we get it down to 2, then that makes life a bit easier. Okay, B, straight away, the bit that's going into the active site is being called the product. Now, the product is going to be smaller than the substrate because the question tells us it's a breaking down reaction. The enzyme is breaking down a larger molecule, which is protein, into smaller pieces. So product cannot be right. So I am actually going to eliminate B completely. That's not right. Okay, other bits are right. Active site, yep. Enzyme, yep. It's this bit that's incorrect. For C, substrate going into the active site, sounds good. Active site pointing to this cleft, brill. Enzyme, yes. To me, C looks like the right answer. I'm going to circle it, but let's just go to D. D, active site, yes. Substrate, definitely not. This is the enzyme, okay? The substrate is this that's going into the active site. Product's correct, but obviously this is wrong. And going back to A, this was wrong as well. So C is our answer for 10. Question 11. Now, this was on also 2017. So this question is one they like to put on the multiple choice paper. It says the diagram shows an experiment to investigate the balance between respiration and photosynthesis. In which tube are photosynthesis and respiration taking place at the same time? So it's actually slightly different question in 2017, but exactly the same diagram. So the balance between respiration and photosynthesis, which one are they taking place at the same time? Well, first of all, if there's just a snail, okay, there can't be any photosynthesis because snails are animals. They don't photosynthesize. So we can get rid of D. Let's eliminate D. Going to A, we've got plant and we've got a snail. However, we've got some black polyphene, which is stopping light from actually getting into the tube to the plant. Now, if there's no light, there's no photosynthesis. So therefore, there'll only be respiration taking place by both the snail and the plant. So A is no. For B, we've got a plant again, which has the ability to photosynthesize. However, just keep a note of this. This is the important bit, really, of the diagram. Again, the light cannot get in. It's being blocked by the black polyphene around the tube. So it'll be respiring, but it cannot carry out photosynthesis due to the fact there is no light. However, in C, we have a plant which can carry out photosynthesis, and we have light able to enter and supply the energy required for that process. So C is our answer for question 11. Question 12. The diagram shows a cross-section of a leaf as seen under a microscope. Which structure is the palisade mesophyll cell? So going through, it helps if you obviously know these structures, you've learnt your diagrams. A, waxy cuticle. That's the bit at the top that protects the leaf from water loss and pathogens and so on entering the leaf. B is actually the palisade mesophyll cell. It's recognizable because it's got these closely packed cells which are jam-packed full of chloroplasts, as symbolized by these little dark cylindrical shapes. C is actually pointing to the vein, otherwise known as the vascular bundle, where you would find the xylem and the phloem. And D, this is the lower epidermis down here. So this is the bottom layer of cells on the underside of the leaf. So B is our correct answer. So as I say, diagrams in biology come up a lot. Learning the arrangement of cells and even organs, say in the digestive system. Make sure you go over those and really get to know what's what. Okay, a data question. We've got a chart used to find a person's recommended mass, so basically what their healthy weight should be. For the data provided about body mass and height, which person would benefit most from the introduction of a calorie-controlled diet and regular exercise? So of our 4 people, A through to D, basically someone on this graph is going to be overweight, or as it's classed here, slightly obese or moderately obese. So the easiest way to do this is to just use the graph for each person and see where they fall. So for A, they are body mass, which is on the x-axis, of 40 and height 155. So that's actually where they would land on the graph. Now they are right where they should be, ideal mass. Okay, so definitely not A. They don't need to be going on a diet. Oh, well, hopefully they've got a healthy diet anyway, but you know what I mean. B, this time we're going up a little bit in mass, we're up to 50, and this person's quite tall. They're 1.75. Now, they're actually low mass, so it might be classed as slightly underweight. However, because the question is asking us about someone who might be put on a calorie-controlled, i.e. lower calorie diet, and take more exercise, B wouldn't be the person that applied to. C, they are 70 kilograms, all right, and 1.8 meters in height. There we go. And they are also, as person A is, in the ideal mass region. So no, it's not you. C, bye-bye C. For D, 90 kilograms, 1.75 kilograms. Where are we meeting? Okay, wait a minute, I've lost my little place. Where are we? 90. 1.75. So they're just creeping into the moderately obese range. Now that's the person who may need to restrict their calories and then take up more exercise in order to lose weight and go back into the healthier sections towards the left of the graph. So the answer is D. 14. The cholera bacterium produces toxins that cause chloride ions to be secreted into the small intestine. How does this affect the water potential of blood in the intestinal capillaries and the intestinal contents? Now, chloride ions are soluble. So if they move into the small intestine lumen, the hollow bit where the food would be, they're going to lower the water potential. And as they're moving from your cells and also your blood through your cells to the small intestine, that's actually going to raise the water potential. Water potential and osmosis are... Oh, I've just circled the wrong one. That wasn't very clever. Try again. Sorry. We're going from raised to lowered. We've got a water potential gradient. Okay, look for a gradient. 2 of these, A and D, are going to be eliminated because actually there's no kind of... It's not like a seesaw where one's up and one's down. We need to see a difference. A gradient is what we call it. So it's always going to go from where there's a higher water potential, which is your blood, to where there's a lower water potential, which is the inside of your small intestine, due to the chloride ions there. And then that means water will move from your blood through your cells and into the small intestine. Hence why one of the key problems with a cholera infection is that you get severe diarrhea due to all that water moving into the small intestine. And of course, that leads you to become dehydrated. Question 15. A student investigates the breakdown of fats in milk by lipase. He's got 4 test tubes labelled A to D. The table shows the contents of each test tube. In which test tube will the contents become acidic most quickly? Now, if you're wondering where this bit's come from, about becoming more acidic, it's because when fats are broken down, there's 2 products. There's glycerol and there's fatty acids. So fatty acids, as the name suggests, will lower the pH and make the solution more acidic. So if we look at our combinations, all of them have got milk, so there's no point taking any notice of this column. That's just the substrate. That's what's being broken down. Now, we're talking about the lipid or the fats in milk. Hopefully, from your knowledge of digestion, you'll know that bile is really important in helping the breakdown of lipids or fats because it helps emulsify lipids. It helps break them down into smaller droplets, which makes it easier for the enzyme to work on. So as C and D don't have bile present, it's going to be unlikely that the breakdown of fat is going to be particularly fast or efficient, if you want to use that word. So now we can just focus on A and B. If we look at the last two columns, they're switched round. So in A, the enzyme lipase is there, but not boiled lipase, whereas in tube B, we don't have lipase, but we've got boiled lipase. Now this part is going on to the effect of boiling an enzyme. If you boil an enzyme, you will denature it. It will lose its shape. The active site, therefore, does not have the correct or complementary shape to the substrate, which in this case is the fat in the milk, and therefore nothing should happen. There'll be no breakdown. So B, as it contains boiled lipase, not lipase that hasn't been boiled, will definitely not allow the breakdown of fat, meaning A is our correct answer. All right, well done. Question 16. A celery stalk was placed into a beaker of blue dye. When the dye reached the leaves, the stalk was taken out and a section was cut. So just here towards the top, just before you get to the leaves, which diagram shows the appearance of the cut end of the stalk. Now, most people do this as a really easy practical. Bit of celery, stick it in some food dye. It doesn't have to be blue. It can be red, whatever, green. Well, no, actually, no, green's a bad colour because it's not very good to contrast against the green of the celery. So, yeah, strong colours like blue and red generally are used just with a bit of food colouring in water. So you might recognise what you're looking at are the xylem. And actually, in the stem, the xylem in a celery stalk look like so, okay, as in B, all right? So all of these little dots, of which there are 5, are the xylem vessels, and they've been stained by the blue dye as it's passed up into the leaves. It's lost by the leaves by transpiration, which is why it gets pulled up through the stem and dyes the xylem as it goes. Okay, right, 17. The table shows the rate of water flow through a tree over a 12-hour period. We've got another data question, this time table. So from 7 in the morning through till 7 o'clock at night, hence 12 hours. What conclusions can be drawn from the table? So what can that data actually tell us? Between 7 o'clock and 5 o'clock, or 1700 hours, the rate of flow continuously increases. Right, let's check it out. Okay, so from 7 o'clock to 9 o'clock, that's an increase of 20 centimetres. Until 11 o'clock, that's another 20 centimetres increase. And then quite a big leap to 110 centimetres. 5 o'clock, another increase of 50. Then when we hit 5 o'clock, sorry, it's okay, question 17. The table shows the rate of water flow through a tree over a 12-hour period. So a data question. What conclusion can be drawn from the table? Between 7 o'clock and 17 o'clock, or 5 o'clock, let's call it. Let's make it less complicated. The rate of flow continuously increases. So this part of the table they're referring to. Now, if you follow through the trend, increase, 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 decrease. So actually from 3 o'clock to 5 o'clock, it actually goes down. So it is not continuously increasing. So A is incorrect. The greatest increase in rate of flow in a 2-hour period is between 11 and 1 o'clock. Well, let's just check. Between 7 and 9, it goes up by 20. Between 9 o'clock and 11 o'clock, it goes up by 20. Between 11 o'clock and 1 o'clock, it goes up by 110, so a big leap. Between 1 o'clock and 3 o'clock, it goes up just by 50, and then actually it starts to go down. So yes, that looks correct to me, and the data does tell us that, okay? That is in the table. Then the next two, just to check that, you know, they are incorrect. There is only one right answer on multiple choice, just to assure you that. Water does not flow up through a tree at night. Well, I've got no idea, because actually none of the data really goes during the nighttime hours. It stops at 7 o'clock at night and it starts at 7 in the morning. I don't really know from the data what's going on at night, so can't draw a conclusion like that. And water flow is affected by humidity. I've got no idea about humidity. This table of data doesn't say anything about the humidity of where this plant is, so it has to be B. Okay, 18. Nearly halfway there, guys. Well done. So another question to do with plants, and this is also to do with vascular tissue, or also known as the kind of vein, if you like, of the plant. We have a diagram showing some of the transport tissues in a plant root. Now Y is pointing to this almost X-like structure in the middle, and Z is the sort of these areas around the outside, and just about tell. They look slightly different from the other types of cell around, just due to the size and so on. Now, in the root, Y, as it's labelled, is the xylem. I always remember it as X marks the spot. It looks like an X, okay? And it's X for xylem. So let's just, first of all, give ourselves a helping hand. Let's go to the table and see what we can get rid of, so that can focus us then on maybe just 2 out of 4 answers. So if we go to the table, tissue Y, okay? We are looking for xylem to be under the name of tissue Y. Now only 2 of them correctly say xylem. So as before, let's eliminate the 2 that must be wrong, because straight away this is incorrect. So, you know, it doesn't matter if the other parts are right. It has to be correct for every single column in the table. Now, between C and D, if you know the job of xylem, you can probably quite easily now recognise that C is the correct answer, that xylem transport mineral ions and water. Z, which is the phloem, which is almost sort of nicely, snugly tucked up inside the arms of the xylem, its job is to carry sucrose. So just check with C and D, you've got just the what it transports, its function is basically mixed up, but C, it's xylem, it carries mineral ions and water, and then phloem, which is Z, is carrying the sucrose, and it also carries amino acids, but that's not mentioned in the question. Okay, 19. We're moving on to transporting animals now. The diagram shows a cross-section through 3 types of blood vessel, not drawn to the same scale. Brill, if you can look at this straight away, make life easy. If you know your stuff, you'll know that V has quite a narrow lumen, but a really thick wall. That's going to be an artery, okay? W, a wider lumen, but a thinner wall. That's going to be a vein. And X, it's going to have a teeny weeny little lumen, and it's only made up of a wall that is 1 cell thick. So from the inside, the lumen, to the outside of the blood vessel, it's just 1 cell in thickness. So X is a capillary. Now, it doesn't ask about V. It just asks about vein and capillary, which is W and X. Hopefully, if you've been able to identify them anyway. So W is the vein. So let's get rid of A. Let's get rid of D. We're looking for W to be under vein, and then X is the capillary. So the right combination leads us to the answer being C. Question 20, we are halfway there. The diagram shows one way the body defends itself against pathogens. We've got a white blood cell. It's engulfing bacteria into the cell, and then these would be destroyed by powerful enzymes that this white blood cell makes. Now, this type of white blood cell is a phagocyte, okay? So the name of the defense mechanism it carries out is, like its name, phagocytosis, meaning to engulf cells, okay? So C is the answer for 20. Right, question 21. We are over the halfway mark. The diagram illustrates changes in air pressure taking place inside the lungs during a complete cycle of breathing. Atmospheric pressure is 101 kilopascals. So that's there on the graph, 101 kilopascals. At which point on the diagram are the ribs beginning to be lowered? Ooh, wonky line. All right, tricky question, because this is to do with pressure in the lungs. Now, when the ribs begin to lower, that's actually going to be decreasing the volume inside your chest cavity or thorax. As that volume decreases, it will do the opposite to pressure. It will increase pressure because that air is getting, if you like, compressed, because the space it's in, the volume it's in, is getting smaller as the ribs move back down and in, or they are lowered, as the question says. So we would expect to see the pressure increasing. So on our little graph here, the only place where the graph is actually showing an increase in pressure in the lungs is A, and it's just starting. And obviously, as those as those ribs move right back into their position, then it's going to further increase the pressure. So A is the correct answer for 21. Question 22. Potted plants are left for a week in a tank of water as shown. Now, I think this is a bit of a rubbish question, but that's just my opinion. It says, why do the plants die? Is it A, the roots do not have enough oxygen? B, the roots do not have enough water? C, the roots have too much oxygen? Or the roots have too much carbon dioxide? Well, seeing as the whole of the roots is submerged in water, I'd say B was a definite no-no. The roots certainly do have enough water, so we can get rid of that. What happens when you submerge a terrestrial plant, basically a plant that should not be submerged in water, is oxygen cannot get to the soil and to the roots therefore. And the root cells are living cells. They need to respire. They particularly need to respire a lot because they need the energy to actively transport mineral ions into the roots for the plant. So A, the roots do not have enough oxygen, is the correct answer. 23. Which statement about aerobic and anaerobic respiration is correct? They break down C6H12O6, also known as glucose. They produce an oxygen debt. They use carbon dioxide. They use oxygen. Okay, well, neither of them uses carbon dioxide, so that's nonsense. Only anaerobic respiration results in an oxygen debt. That's the lactic acid that needs to be broken down. They both use oxygen. Well, if that was the case, then anaerobic, which means without oxygen, well, it wouldn't be anaerobic. So that's not the case. The answer is A. They both break down glucose. That's the substrate. That's what's needed to release the energy for the cell. 24. The components of a reflex arc are shown in the flow diagram. Which component is responsible for ensuring that the nerve impulses travel in one direction only? Again, not the best question really, because actually nearly all of it is responsible for allowing the nerve impulse to go in one direction. But I'm imagining CIE want you to pick synapse, okay? The synapse, because neurotransmitter is only released on one side of the synapse and the receptors are only on the far side, it can only go in one direction. So the answer is synapse. What is a function of the liver? Converting bile to urea? No, the liver actually makes bile. Converting urea to amino acids? Nope, other way around. Deamination of amino acids? Yes, it gets rid of that nitrogenous bit, the bit containing nitrogen on the amino acid, which is what deamination means. And that's how we form urea, which we get rid of via the kidney in the form of urine. Deamination of CO2, right, well, definitely not. That's nonsense. Whoops. Okay, so the answer is C for 25. 26. Which statement about light receptors in the retina of a normal human eye is correct? The cones only work in dim light. Definitely not. They'll only work in bright light. The rods are found in the fovea. No, the cone cells are mainly concentrated in the fovea. There are 3 types of cone. Yes. Trichromatic vision is down to the fact we've got 3 types of cone to see different wavelengths of light, which then gives us the colours. And there are 3 types of rod. No, only 1 type of rod. They like asking that question about how many rods and cones there are. Again, in 2017, there was a similar question, but it was presented as a table rather than a list of statements. Okay, a patient is injected with adrenaline. Which graph would show the expected changes to pulse rate and blood glucose concentration? Again, the same question was asked in 2017. However, it was done in a table format. Both your pulse rate and your blood glucose concentration will go up due to adrenaline. Your heart's going to beat faster, which means a faster pulse rate, and you're going to want more glucose in your blood for extra respiration, for fleeing a predator or fighting a predator. It's the whole thing to do with the flight or fight response. So the only graph which shows an increase in both pulse rate and with blood glucose conc is graph C. 28. The diagram represents a synapse in the brain involved in the perception of pain. Which labelled molecule represents heroin? So here's the synapse. between the end of one neuron and the start of the next. Now, again, I don't think this question is particularly brilliant, but hey, we'll give it a go. What this is supposed to represent is what we call the receptor site, so where the neurotransmitter would bind on the far side of the synapse. Now, if you look at your 4 different molecules, which are represented by shapes, only A looks like it would be able to bind because it has a complementary shape to the shape of the receptor site. So the other 3 would not fit, and therefore that would be incorrect. 29. A human zygote is a diploid cell. Now, in case you don't know what a zygote is, it's that initial cell formed after the egg cell and the sperm cell come together in fertilization when their nuclei fuse. So which statement about a human diploid cell is correct? They do not have a nucleus. No, most do, apart from there are a couple like the red blood cell, which as it matures loses its nucleus, but that's a very extreme specialization to carry more oxygen. They fuse to form gametes. No, that's backwards, isn't it? Gametes fuse to form diploid cells. The nucleus contains a single set of chromosomes. No, because that's what haploid means when it's got half the normal number of chromosomes, and that's what a gamete would have, a sperm or an egg cell. So D, the nucleus contains 2 sets of chromosomes. Yes, and that word di means 2. Okay, question 30. We are 3/4 of the way there nearly. Which feature allows the sperm to dissolve the jelly coating of the egg cell? So you need to know your sperm structure really to answer this question. Right at the tip of its head, there is a little pocket containing powerful digestive enzymes, and that's known as the acrosome. The flagellum is the name for the tail part of the sperm, so that's not to do with dissolving the jelly coating, that's just for movement. The mitochondria provide the energy for the flagellum to move. So again, not to do with breaking down the jelly coating, and the nucleus is just the storage of the father's genetic material, his DNA. So again, it has no role in allowing the sperm to get into the egg cell. So the answer is A. Question 31. Which statement about HIV is correct? A, it causes an increase in the number of lymphocytes. B, it is caused by bacteria. C, it increases the ability to produce antibodies. And D, it is transmitted through sexual contact. If it's easier, I keep saying this to you guys, to eliminate the answers that are rubbish or nonsense, whatever, is the best way to sort of work through the question a lot of the time. So first of all, an easy one we can get rid of. HIV is the human immunodeficiency virus. The V stands for virus. So for B, that's definitely a no-no. It's not caused by bacteria, it's caused by a virus. So B can be eliminated. Now, A and C are both saying they're going to increase, I suppose, your immunity in a way. They're going to increase the number of lymphocytes, which make more antibodies. And this one is also saying the same thing. We're going to get more antibodies. Now, that would actually be the opposite of what HIV does to the body. HIV actually decreases the number of lymphocytes, and therefore it makes it harder for your immune system to defend you against pathogens that would cause diseases, or even just, common cold and things like that we all pick up. Now, the one that is correct is it can be transmitted through sexual contact. That is not the only way. Obviously, sharing of needles, things like that can also be a way it can be passed from one person to another. But certainly, it can be transmitted through sexual contact if there is no contraception used that's a barrier method that would prevent that kind of transfer, if you like, of bodily fluids. 32. The diagram shows the chromosomes in the nucleus of a cell that divides, go the right way, by mitosis. Okay, which diagram shows the chromosomes in the nucleus of one of the daughter cells produced? Now, mitosis produces an identical copy of a cell, so it conserves the chromosome number. It means it keeps it the same. So easy, a little bit of counting. Let's just count up how many chromosomes we've got to start in our parent cell. 1, 2, 3, 4, 5, 6, 7, 8. All right, 8 chromosomes. Okay. Now, in A and B, they've got basically the same chromosome number. They've just drawn them slightly in different positions. So there's only 4 chromosomes. That can't be right. Mitosis does not have the chromosome number. Meiosis does, the one without the T, but this question is about mitosis. So let's look at C and D. Now, if you count carefully, we've got 1, 2, 3, 4, 5, 6, 7, 8. All right, that's right. D, to the eye, might look like a similar number. However, crafty CIE, 1, 2, 3, 4, 5, 6, 7. No, 8 has not become 7. Somewhere a chromosome's been lost, and that's not good. So C is the correct answer. 33, we're into the home stretch now. What happens as a result of meiosis? So there you are. This is the cell division without the T. Okay, this is the one that produces gametes, sex cells as they're also known. Right, does meiosis produce diploid cells? It does the opposite. It produces haploid cells, which have half the normal number of chromosomes. Does it produce both diploid and haploid? No, nonsense. Are genetically identical cells produced? No, because the whole point of meiosis is to produce cells which are different from one another. The whole idea of gametes is about variation in a species. So our offspring aren't the same as us. Their offspring are different from them, and so on. So we are looking to increase variation, which aids in survival of the species. And finally, are haploid cells produced? Oh, yes, they are. Okay, so that's the answer to 33. 34. Why is the allele for sickle cell anemia common in some parts of the world? So when you were taught this bit about variation, if you like, and also selection, you may remember that in some areas of the world where malaria is prevalent, sickle cell anemia, if you have one faulty allele, you're a heterozygote, it actually gives you protection against malaria. Now, it's a wordy question because they've just jumbled up the words in just slightly different places in the sentence to change the meaning. So, does malaria protect against sickle cell anemia? No, it's the other way around. Does sickle cell anemia, is it caused by malaria? No. Malaria is caused obviously by it's a type of protist called Plasmodium. So it's actually passed to you by a female mosquito, which feeds on blood. Sickle cell anemia is purely down to genetics. You can't catch it from another person. You can't get it via an insect bite or an animal bite, whatever. Okay. Sickle cell anemia is transmitted by mosquitoes. No. Sickle cell anemia is passed on via your parents. It's genetic. And the sickle cell anemia allele protects against malaria. There it is. Last one in the lineup. It's D. Okay, question 35. Which statement describes how a species becomes adapted to its environment? Again, it's so wordy, so just take your time. Are genetic similarities able to give rise to different genotypes which may have a reproductive advantage? Well, it kind of contradicts itself. It's saying if we're similar genetically, we'll end up with different genotypes. That doesn't sound right, does it? Okay, genetic variation will give rise to different phenotypes which may have a reproductive advantage. Now that one sounds good, so I'm going to ring it. If you've got different DNA to someone else, you may have a different phenotype. So you may, for example, have brown hair and someone else might have blonde hair, and that's down to genetic variation. You inherited different alleles from your parents, who inherited different alleles from their parents, and so on. Phenotypic, so this means about your characteristics, are similarities give rise to different genotypes which may have a reproductive advantage. Okay, it's back to front. It's your genotype that gives rise to your phenotype, so that's why that one is incorrect. And again, for D, phenotypic variation gives rise to different phenotypes which may have a reproductive advantage. It's your genotype, which is your combination of alleles that you inherit, that actually then dictates your phenotype. So the wording is not correct. But these wordy ones are horrible, really. I think they're just as bad as the one with all the table ones where you've got to get everything lined up in the right order. Okay, question 36. Decomposers play an important role in the carbon cycle. They release carbon dioxide. Which process is responsible for this? Don't ever forget that a decomposer is a microorganism. It's a bacteria or a fungi, okay? Or a fungus, sorry. What these organisms are doing is they are basically respiring, and when they carry out respiration, they break down whatever it is they're decomposing to get energy, because they are living cells, and they release carbon dioxide. So the process is respiration that they are carrying out. 37, last few now. Which term describes a group of organisms of one species living in the same area at the same time? So we're on to our ecology part of the syllabus now. If you belong to the same species and you're living in a certain area, I don't know, like a wooded field or something, at a particular time of the year, you are known as a population. Don't get it confused with a community. A community is the next step up, where you've got a group of different species living in the same area at the same time. 34. The diagram shows a bacterial cell. Which part of its structure is particularly useful in genetic engineering? So if we just go around what everything is in the structure, A is the cell wall of the bacteria, B is just going to the cytoplasm, C is the genetic material. Remember, it's not enclosed in a nucleus because these are prokaryotic cells. They don't have a nucleus. D, although it's probably a little bit out of scale, it's massive compared to the rest of the cell. These are actually very small structures. This is called a plasmid. And this is this ring of DNA that bacteria may have several of these, and that can actually have enzymes inserted into it from, this can have genes inserted into it from other organisms such as humans, and we can actually get those bacteria to make our proteins for us, such as enzymes or hormones like insulin. So D is the correct answer. Last 2 questions, we're nearly there. What can be managed sustainably? Taking coal from a coal field, taking gas from under the Earth's surface, fracking, taking oil from an oil field, taking wood from a woodland. Now, sustainability means we can kind of try and replenish or replace what we're using and try and keep it at an even keel so we don't run out of something. These 3, we cannot replace coal and gas and oil at the rate we use them. They take millions of years to form as fossil fuels. However, what we can do, don't know how good we are at doing this to be fair, if we're taking wood, we can be planting trees to replace those that we've cut down, okay? But do bear in mind, these trees take a long time to grow to the maturity of the trees that we're cutting down, so there is a slight problem there. 40. Large areas of tropical forests have been cleared to grow a monoculture of palm oil plants. A monoculture, as the name suggests, is just where you're growing one type of plant only. Mono one. Think monobrow, one eyebrow. Which effect will it have? Which effect will this have on the ecosystem? Will it cause the use of fossil fuels in the area to decrease? The use of pesticides in the area will decrease. The variety of species in the area will decrease, or the variety of species in the area will increase. Okay, well, if you're growing one type of plant and basically you're going to clear anything else that's growing there, you're also going to affect the animals, because animals are not going to have the same food sources that were there previously. You are going to be massively decreasing the number of species and the variety, if you like. It's what we call biodiversity. You're just trying to grow one type of plant because it makes it economical for the farmers to just grow one type of plant. It's not good for any other species, which will be cleared out or killed in the process. Okay, guys, I'm going to put the mark scheme up for you so you can just check through. I hope you managed to get most of those right yourself as we went through. Have a look at the grade boundaries for this paper, see what you scored out of 40, and you will get an idea of what grade that would have equated to if you'd sat this paper. Well done.